So today we're working on an experiment to study anaerobic respiration and the factors that can change the rate at which it occurs in yeast. What we have are a series of fermentation vessels that you can push all of the air out, and then on the side of them you can see how much gas bubbles have been formed by the yeast. So there are 7 over here. And in your supplement manual, there is a table explaining what reagents you should place into each one before you add the yeast. So in the large flask is our yeast. In vessel number 1, we added 2.5 milliliters of a sugar solution, 5%, and we also added 5 milliliters of water. In the second one, we've added 5 milliliters of magnesium sulfate, 2.5 milliliters of the sugar solution. In the third one, we have added 0.5 milliliter of sodium fluoride, 2.5 milliliters of sugar, 5%, and 4.5 milliliters of water. In the fourth one, we have added 5 milliliters of sodium fluoride, 2.5 milliliters of sugar, 5%. In the fifth one, we have added 2.5 milliliters of sodium pyruvate, 2.5 milliliters of sodium fluoride, and 2.5 milliliters of our sugar solution. In the sixth one, we have added 2.5 milliliters of sodium pyruvate, 2.5 milliliters of sugar solution, and 2.5 milliliters of water. And in the last one, we have 2.5 milliliters of sodium pyruvate, 2.5 milliliters of our sugar solution, and 2.5 milliliters of water. The next thing we have to do is pour the yeast solution into each of these vessels in such a way that we get all of the air out and the vessel is completely filled. And then we'll cover the end of it with parafilm and pop a little hole in the parafilm. So what you can see is that Zeke is tipping that reaction vessel to allow air bubbles that are in the skinny part of the tube to come up into the wider part of the tube. And you just tip it back and forth gently until you get all the air bubbles out. And it may take a few tries.
I forgot to mention reaction vessel number 7 is our negative control, and we're adding only water to it to fill it to the top, not yeast and sugar as with the other tubes.
All right, now that it is completely filled and there are no air bubbles, he's taking a piece of parafilm and securing over the opening to make sure that nothing can evaporate from inside and that no air can backfill. And then he's going to take a small capillary tube to poke a small hole in the parafilm. Now we'll repeat this step with all of the other tubes, just adding the yeast, putting the parafilm wrapper on them, and poking a hole. And then we'll place them into this incubator right here, which is set to 40 degrees Celsius. So the time has passed and we've taken them out of the incubator. And we've lined them up in order again, and you can see the varying amounts of bubbles in each of them. We measured them, and from the distance to the top of the tube to the distance of the top of the bubbles in each of these tubes. When you think about the results, think about which of the reagents we added to each of these tubes besides just the sugar, the water, and the yeast solution. Which of these would act as a promoter or an inhibitor based on what you know about anaerobic respiration and the cycle of chemical changes that have to occur? Sodium pyruvate mimics an intermediary in that cycle, so we would expect it to behave as a promoter. Magnesium sulfate also we might expect to behave as a promoter. But notice what happened to the tubes with sodium fluoride. That was tube 3, 4, and 5. They have less growth than 6 and 7 do. 6 and 7 were our controls. And as you can see, number 4 had almost no growth, no bubbles hardly. That is because it had 5 milliliters of fluoride added to it. Number 5 did have some growth, but not as much. And the reason is that there was less of the inhibitor and also some promoter added. Number 3 has only a small amount of inhibitor and no promoter to counterbalance it. You should be able to complete the rest of the questions in the lab report based on this data.